Okay, moving on to dihybrid crosses. Dihybrid crosses are one of the more challenging crosses that you will have to do for Bio 30. They do get more challenging when you go into university levels, but for Bio 30, I'd say this is one of the toughest ones. So before we get into dihybrid crosses, Mendel's second law of genetics is known as the law of independent assortment. Basically what it's talking about is that you do have many genes that exist on your chromosome. Now, when you're creating your gametes through the process of meiosis, those genes are going to segregate independently. So they're going to separate independently of others. So one gene will not affect another gene, is essentially what it's saying. And then you can read the definition that's more scientific. So, dihybrid crosses are super important. I like to call this two Punnett squares in one. So you're going to take two monohybrids and you're taking two genes basically, and you're seeing the probability that you will get a certain offspring with two traits and how those two traits relate to each other. It is always important to reduce your ratios to your lowest common factor. Again, a lot of students forget to reduce. So if you have a 2-to-2 ratio, it's 1-to-1, or a 2-to-4 is a 1-to-2 ratio. So reduce. So our first example, Mendel. Sorry, let's get my pen out here. Mendel crossed a plant that had two alleles, which were both homozygous for tall and green. So if you remember back from the monohybrid video, tall was dominant and green was dominant. And you can look back on the first page of these notes packages for your chart so that you can see which is dominant and which is recessive. So homozygous tall, so big, big, and homozygous green, big, big, with a plant that is homozygous for short and yellow. So basically what it's saying is you have a purebred dominant for two alleles crossed with a purebred recessive for two alleles crossing together. So it says write the genotypes of the parents. So what do we want to use for tall? What letter? I don't know. Let's stick with B. Okay, big B, because it's homozygous, so the same. And then we have green. Well, let's use G for green. Okay, big, because it's also homozygous for green, which is the dominant trait. Now we're taking the exact opposite, homozygous, but for short and yellow. So you should have little, 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 little. So this is representing one gene. So you have one gene, and then this is your other gene. One gene, this is your other gene. So you have one allele, two allele, one allele, two allele. Okay, so I wish this one was on the same page, but I was trying to save paper. So we had big B, big G, crossed with little b, little g, little g. Okay, so it says create a Punnett square for the F1 generation. So this is your parental generation 1. This is going to be our filial generation 1. Okay, so when you're creating Punnett squares for a dihybrid, you must remember to FOIL. Okay, so FOIL used to be a math term that a lot of students used. I don't know if it's used so much in math anymore, but the letters of FOIL represent what you're doing. So you're taking the first alleles, the outside alleles, the inside alleles, and the last alleles. Okay, so I'm going to show you what this means. Okay, I have to determine what I'm going to be drawing up here, 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 and here. Let's start with our dominant one. Now, this should be pretty self-explanatory because they're all the same alleles, but first, so you're taking the first allele out of both genes. So first and first. So you're taking big B, big G. Okay, outside is talking about the first allele and the last allele. Now you should see that this is all going to be the same. Inside is talking, now I'm going to change colors so you can see the different colors. Inside is talking about these two. And then last is talking about the last alleles. So this one's pretty easy because they're all the same alleles. So if I look at this one, you can see that you're all going to be little b, little g. Well, I'm getting quite angly here. Okay. So now let's look at what would be your outcome. So in my first box here, remember, dominant takes over. Your B alleles should stick together and your G alleles should stick together. So I have a big B and a little b. So you get big, little, and then you get a big G, a little g. Big G, little g. Okay, next one. Big B, little b. Big G, little g. So since all of these are the same and all of these are the same, you should get all of the exact same. Okay, and we're just going to shortcut it. All of these are going to be the same. So it says, determine the probability of genotypes and phenotypes in the F1 generation. Well, all of them are the exact same. So all of the black marker that I wrote, that's talking about your probability of your offspring. So 100% for my genotypes, big B, big G, little g. So what does that mean? The B's represented height, so whether it be tall or short. So this is a heterozygous, dominant takes over. So this should be tall, and then you have a big and a little. So the big is dominant, it takes over, so this was green. So we have 100%, those look like sixes, sorry. Tall, green. Okay, I want you to try D by yourself. I'm going to skip ahead and I'll post the filled in version so that you can see if you've done it right. So I'm going to do number 2 here. So you are crossing a plant that is heterozygous for tall. So I'll stick with B's, heterozygous, big little, with homozygous round seed. Okay, well, is round dominant or recessive? You can read the rest of the question and it might give you a hint on that, or you can use the previous chart. Okay, so I'm just going to read the rest of it because this is what you're going to get on your test. Because it might not give you a chart, it might just give you this and you have to assume. So it says the plant is homozygous tall and heterozygous round seed. So heterozygous means big little and it's telling you that it's round. So that means that dominant should be the rounded seed. Okay, so homozygous round seed would mean big, big. Then you have homozygous tall. So homozygous tall, tall was dominant, so you have big B, with a heterozygous round seed, so big, little. Determine the phenotypic and genotypic ratio of the F1 generation. So P1, what two individuals am I crossing together? So this, so I get big B, big R, being crossed with big B, big R, little r. Okay, now I have to create my lovely chart. Well, I don't know how straight this is going to be, and I'm sorry if it's not 100% straight. I usually do this on the whiteboard, and I've got tons and tons of room on the whiteboard. Not so much on here. Okay, so foil. Let's start with our first parent first. Big B, big R. Okay, O is next, or outside, big B, big R. Inside, little b, big R. and last. Okay, so there's our first. So I'm going to create my lines going down. And I'm going to do my next one. So, foil, remember, F-O-I-L, firsts. Big B, big R. Outside, big B, little r. Insides, and last. Okay. Now you've just got to cross them. Okay, and I want to get rid of some of my lines. I'm sorry. A little messy. Okay. Let's try and figure out what we've got now. So in here, you get big, 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 big. So it's homozygous dominant for both of them. Okay. Big, 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 big. Oh, it's the exact same. Yes, I do recommend that you write them both so that you don't forget counting them in the end. So you have a big B and a little b, big R, big R. And then this one should be the exact same. Actually, it's not. Little b, oh no, big B from over here, little b, big R, big R. Okay. Big B, big R, little r. Big B, big R, little r. Do you notice a trend? First two are the same in all of these. Big B, big R, little r. Big B, big R, little r. I highly recommend that you pause the video so that you can try the rest of them and then check to see if you did them right. So big, 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 little, big, big. Just making sure I did it right. I'm doing these pretty fast. Big, little, big, big. And last but not least, big, 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 little, big, 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 little, big, little, big, little, and big, little. Okay, there's your Punnett square. And yes, you need to do this before you figure out probability. On a test, you'll be given this, and then you have to tell me the probability. So you would have to do this on a separate piece of paper. So, genotypes. Okay, genotypes are your letters. So I highly recommend that you use highlighters for this. Okay, now, because you have to count them. So let's start at the top here. Big B, big R, big R. Find all of your big bigs and highlight them. There's another one. No. Oh, no. Oh, there's another one. Here's another one. Big little, no. Big little, no. Big little, big. Oh, but then heterozygous, so no. So I only have 4 of these. So then what you're going to do is you're going to write 4 big, and then go to your next highlighter. Okay, so go to your next genotype. So we have big, little, big, little, big, okay. Big, little, no, not big, not big. So you're looking for 2 big bigs at the end. So there's another one, and there's another one that's the exact same. So for this one, You would also have 4 big, little, big, big. Then you go to your next one. Change colors. Okay, big, 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 little, big, 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 little. No, 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 no. Oh, there's another 2. Do you see a trend that's happened? Not all of your Punnett squares are going to be like this. So we had 4 big, big, big littles. And last but not least, you don't have to highlight these if you don't want to, but my OCD kicks in. Big, little, 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 big, little. So I have 4 big, little, big, little. So heterozygous, heterozygous. So I have a 4 to 4 to 4 to 4 ratio. Remember to reduce. So I am okay if you show me what the 4 to 4 to 4 is up here with the correct genotypes, and then you just reduce it like this. Okay? But you have to show me this up here first. Okay, now you have to do phenotypes. Okay, so these are what's being expressed, so what you see. Okay, so now we have to start counting again. So I'm going to get a highlighter out. Okay, I love highlighters for this unit because it's just so much easier to work with and see. Okay, so I'm going to start with this guy. Big, 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 big. So the B's represented tall, so I have a tall, R meant round, so tall, round. Okay, go to your next one. Big, little, so tall, big, big, round. So this is another one of the same. I have big, big, tall, big, little, round. Okay, my next one. I have big, little, tall, big, little, round. So for this one, and this Punnett square, all of them, we have 100% tall, round seed. It may not necessarily be tall, round. Okay, you might have 16 different genotypes here, and then you have to list them all. You might have 9 or 10 different phenotypes, and yes, you have to list them all. Okay, try number 3 on your own and check to see if you did it right. The next one talks about incomplete and codominance. That'll be next week on the next video lesson. Please, please, please try the 2 on your own before you check your answers, because it's very important to practice before you do your summative assignment. Good luck.